Okay, hello everybody. So during the classes Massimiliano was teaching, basically introduced like a non-equilibrium description, right, to characterize the energetics of chemical reaction networks. But the basic assumption was to track down all the elementary reactions, all the reactions that were out of equilibrium. And so when dealing with very large systems, with a lot of reactions, with a lot of species, how can we apply that? And the point is that applying that formulation is very hard because most of the time we don't have enough information to track down all these reactions, to track down all these species. And the way to go around this problem, I mean, the way we found a way to go around this problem is by developing a sort of coarse-grained approach, which is like similar to circuit theory in electronic circuit theory, by developing such a coarse-grained description, which is still thermodynamically consistent. So to introduce you to this kind of problem, let me take the long way around. So as also Pablo was mentioning last week, thermodynamics is a very old theory. It was developed originally in the 19th century to characterize the energetics of macroscopic steam engines, which operate between equilibrium states. So it was originally developed as an equilibrium theory. And the second law basically introduces a new quantity, the central production, which quantifies the dissipation, so how much energy I lose along each transformation, and states that this dissipation must always be positive. And together with the understanding about the energetics of these engines that came with thermodynamics, came also the question about the macroscopic origin of thermodynamics itself, which was explained in terms of statistical properties of molecular system, if you want, both at the classical and the quantum level. But since thermodynamics was developed as an equilibrium theory, originally also these approaches were developed at equilibrium. But like as we were taught during all this school, most processes and life itself is out of equilibrium, right? So people starting trying to understand how to extend thermodynamics beyond equilibrium. And the first idea was like to consider systems close to equilibrium, because thermodynamics was developed as a closed theory in terms of thermodynamic quantities only. And to develop a non-equilibrium theory that is nowadays known as irreversible thermodynamics, which was still closed in terms of thermodynamic quantities only, the only kind of processes that people were able to characterize were processes close to equilibrium. And this was also understood in terms of statistical properties using linear response. And it's only by the end of the 20th century, beginning of the 21st century, that a full non-equilibrium theory was developed. And the trick to go beyond processes close to equilibrium was to build a thermodynamic description on top of a dynamical system, and to use the dynamical system to track down the energetics, as Massimiliano was explaining in the previous classes. We have a dynamical system, and then we build a thermodynamic description on top of that. And since originally the dynamical system that was used was a stochastic one, the theory is nowadays known as stochastic thermodynamics, but it can be developed also for deterministic processes, for deterministic chemical processes, as Massimiliano did in the previous 3 classes. But the point of the theory is that we need to track down all the processes that are out of equilibrium. And for us, it means all the reactions, all the elementary reactions. So you can already imagine that the standard systems that we can study with this kind of theory are small systems, where there are few processes that are out of equilibrium. And so the challenge is to understand how to characterize the energetics of systems where there are a lot of processes, a lot of degrees of freedom that are out of equilibrium, and where most of the time we don't have the models that track down all these degrees of freedom. A prototypical example is active system, where you have these swarms of bacteria swimming around, harvesting energy at the level of the metabolic network, and then pushing macroscopic gears and performing macroscopic work. So the idea is that you have so many layers, you have degrees of freedom scattered all over many scales, which is very hard to track down the energetics to track down all these processes that are out of equilibrium.
Yeah, I just had a question about the comment that you made about deriving this from dynamical systems. Yeah, so you were, so you were talking about that, right? And I remember Massimiliano showed that. One thing that struck me in that is that a lot of the things that we did seem to rely on the existence of a Lyapunov function in the dynamics, right? For instance, we had this Gibbs free energy, which always, decrease along a trajectory. I'm just sort of wondering, is that necessarily a requirement? For instance, you can have lots of dynamical systems where it's not obvious if there is a Lyapunov function, and what parts of these?
So I would say that if the system is detail balanced, meaning that there's a well-defined equilibrium, you probably have a Lyapunov function, because these are dissipative systems. So they dissipate free energy, whatever, and they relax down towards equilibrium. So there's like a strong attractor. And for those systems, you have a Lyapunov function. But like also for chemical processes, when you have a steady state, which is not an equilibrium one, you don't need to have a Lyapunov function. It's not granted at all.
Got it. Thank you.
No problem. So in the talk of today, I'll try to show how to go from the description that Massimiliano introduced in the previous classes towards something that can characterize also processes with many degrees of freedom. Okay? And I'll do that for chemical processes with an application to central metabolism of E. coli. Okay? So let me just try to sum up what Massimiliano introduced in the previous classes, just to be sure that, we remember the basic stuff that I need to use for the next step. So the idea is that we have a rate equation describing how the concentration Z of the chemical species change in time. And we have a first term, Sj, which accounts for the effect of the chemical reactions. And our systems are in general open, so we have I, a term accounting for the exchanges between the system and the environment. If you think about a metabolic network, you can have like the exchange with the environment, including fuel, like nutrients, waste, and then you can imagine that basically metabolism is a huge engine harvesting the energy from the interconversion of fuel into waste to power up the interconversion of a low energy molecule, ADP, into a high energy molecule, ATP. And I mean, the model Massimiliano was using to, as a model system to apply the theory, in a way, if you want, is a very minimal and naive representation of a metabolic network. You have an enzyme catalyzing the interconversion of fuel into waste, coupled to the interconversion of a substrate into a product. And you can think of it as ADP into ATP. And we characterize the topology, the stoichiometry of the reactions using a stoichiometric matrix. And this is exactly the stoichiometric matrix that Massimiliano wrote in the previous classes for this specific network, where each column is telling us the net variation in terms of number of molecules for each species along a specific reaction. Right? And as I was saying before, the way to construct a thermodynamic theory is by, first of all, assuming that our dynamical system is tracking down all these elementary reactions that are out of equilibrium. And then with the local detail balance assumption, which is a consequence, if you want, that closed systems must react towards an equilibrium, we can relate the fluxes of each reaction to the corresponding variation in terms of the Gibbs free energy, the delta G of the reaction. So the dynamics and the thermodynamics are not independent, they are coupled together. And that's what allows us to write down thermodynamic quantities like the entropy production, which can be written as the sum over all reactions of the delta G of the reaction, if you want the thermodynamic force, times the corresponding current. Okay, and as Massimiliano was showing again, these currents and forces are always aligned. So if the delta G is negative, the reaction proceeds in the forward direction. If the delta G is positive, the reaction proceeds in the backward direction. And the entropy reduction is always positive and quantifies how much free energy we are burning, we are losing. And these are just the delta G of the reactions in the network he used as an example. Right. So in theory, with all of this, we could apply the theory to whatever chemical process which is described by the deterministic rate equation. But if we think about applying, yeah, okay, sorry, forgot about that. And basically, you can answer, I'm coming in a sec, the same kind of questions people were asking about macroscopic steam engines. How much energy do I need to interconvert ADP into ATP? How efficient this process is? Can we derive sort of guidelines to optimize it? And things like that.
Francesco, so when we do calorimetry, are we measuring this T?
Not only that.
Okay.
So if you want the entry production, there are different contributes. One is heat. The other is also the exchange of entropy between the system and the environment. So with calorimetry, if the system is open, you don't get only, I mean, you just get heat, which is one of the contributions to the entry production.
Okay.
Yes, yeah. Sorry, the notation is slightly different from what Massimiliano used. Okay, but then if you think to apply this to metabolism, okay, there are so many reactions, so many species, and we don't have the models to track down all the enzymatic reactions, if you want, all the elementary reactions. We don't, and even if we had the models, there are so many species, there are so many reactions that the computational effort to characterize dynamics and thermodynamics of these kind of processes is huge. So maybe we don't want to approach this, you know, with a brute force approach. Maybe we want to find a better way, a different way around the problem of characterizing all elementary reactions or elementary processes that are out of equilibrium. And the trick, the solution we found is basically to characterize a complex network in terms of modules, subparts that are then coupled together, and the reactions inside these modules are coarse-grained into effective reactions that are then coupled between species. Now, to explain this, I'm not going to use metabolism because it's too complicated and I'm not a biologist. So let me use an example. Let's pretend that we are dealing with the network here on your left. And let's pretend this is like a very complicated network with too many reactions, with too many species that we cannot apply the thermodynamic description Massimiliano developed in the previous classes. So what's the trick? The idea is to split the set of all reactions that we have here on the left, the full network tracking down all elementary reactions. We want to split them in subset of reactions that interconvert a subset of species. The label M stands for modules, because the idea is to group subset of reactions and characterize them in a simpler way in terms of these modules. And the species that are interconverted by only the subset of reactions can be split into two sets. The internal species, this is important, are those that are interconverted only by the subset of reactions. Like here, you see the species that are like enclosed in this sort of blue box are only interconverted by these reactions. Here, the species in green, enclosed in this green box, are only involved in the reactions here represented in green, etc., etc., etc. So the internal species of a module are unique of that module. They are not shared with the other modules. Then we have these terminal species that are instead those that are shared between different modules. They are interconverted, for example, here, both by the blue reactions and the green reactions. Question? Yeah, that's a problem. You need to be sure of that. And for example, for metabolic steps, you need to be sure, for example, if you want to coarse grain the enzymatic steps, right? You need to be sure that that enzyme is not shared with other reactions. I will comment on this also a little bit further because this is a crucial point. And I'll show that by analyzing this specific orange module. But I mean, the problem is there. I mean, if you want to do this experimentally, you need to know that the species that are hidden away inside your module are unique of that module. But we need some, I mean, if we want to simplify a problem, we need some assumption. Otherwise, like, I mean, we need to use the original theory and we are back to step 1 or 0, depending on. Okay, so let me like stress again the idea. We have a subset of reactions and we split the species internal. And if you want, this is also the same kind of nomenclature Massimiliano was using. The species internal of the module are unique of the module. And then we have terminal species that are shared between different modules. And if you want, the terminal species are the equivalent of like conductors coupling different electronic devices. And the point is that we want to characterize each single module in terms of the terminal species only. Okay, we want to forget about the internal structure, the elementary reactions of the module, and we want to convert whatever complexity is there into just effective reactions between the terminal species. And I'm using this expression of effective reactions exactly as Massimiliano was saying, that we could use emergent cycles to derive effective reactions between species that are exchanged. So this is just to like anticipate that the trick here is to use the cycles Massimiliano introduced the other day. So let's see how we can do that. So imagine this is exactly the green module that was also in the other slide, and with different symbols. This is also the same network Massimiliano used as an example in the previous days, okay? And imagine you want to characterize this independently of the rest of the network. So some species, F and W, if you remember, were exchanged with the environment, okay? The other species, NA and NB, are coupled to the rest of the network. So I use different symbols to like hide away either the environment or the rest of the network. And if you want, we can write down a rate equation for the species Zm, the species of the module, including both internal and terminal species. And the rate equation is exactly the same. The point is that this exchange current now accounts for both the exchanges with the environment and the effects of the rest of the network on these two terminal species. And we can split the rate equation as like for internal and exchange species, here for internal and terminal species. I use Q for the internal species and I use P for the terminal species, okay? And this is the sub-stoichiometric matrix for the internal species, and this is the sub-stoichiometric matrix for the terminal species, as Sx and Sy were the symbols Massimiliano used for internal species and exchange species, respectively, right? And the objective, as I was alluding to, is to derive a closed equation in terms of terminal species only, by hiding away, by coarse graining, all the complexity that is inside the module. How to do that? Okay. Okay, let me explain it, because the trick is exactly the same as Massimiliano used, with a little difference. So the assumption, again, we need an assumption to simplify the problem. And the assumption is that the internal species relax very quickly to the steady state imposed by the terminal species. The terminal species can change in time, but the assumption is that for whatever concentration of the terminal species we have, the internal species relax very quickly to the steady state corresponding to the concentration of the terminal species. So there's a difference in time scale between the evolution of the terminal species and the evolution of the internal one. And this kind of time scale separation is quite easily obtained when there are differences in the abundances. So imagine that we have a high concentration of the terminal species and a low concentration of the internal one. This is enough to create a time scale separation, because on the same time scale that the concentration of the terminal species, which are very large, change very little, the concentration of the internal species, which are smaller, can dramatically change and reach a steady state. That's the idea. So if we consider that there's this instantaneous relaxation towards a steady state, then we can write the rate equation, sorry, the reaction currents evaluated for the concentration of the terminal species and the steady state to which the internal species have relaxed instantaneously or almost instantaneously, of course. Well, this is like the steady state current of the dynamical rate equation for the internal species. And so we can write it as a linear combination of cycles, the same cycles Massimiliano introduced, right? Because J must be a right null vector of the sub-stoichiometric matrix for the internal species. So we can write it as a linear combination of cycles, exactly like yesterday, right? Exactly with the idea of saying, okay, we are a steady state, so the current is a linear combination of cycles, and we just have to, and these coefficients of the linear combinations are in general functions of the concentrations of the terminal species. And when we plug this in the rate equation for the terminal species, now this is the same kind of expression Massimiliano was showing yesterday. So the product between the sub-stoichiometric matrix for the terminal species and each cycle gives you the stoichiometry of an effective reaction between the terminal species. Because the cycles are those sequences of reactions which upon completion don't change the abundances of the internal species. But the net effect is to change the abundances of the terminal species in this case, or the exchange species until yesterday. And so by collecting these vectors, so these are vectors, right? Because they are the product between a matrix and a vector. By collecting them into a new matrix that we call SEM, encoding the stoichiometry of these effective reactions, we can write down the rate equation in this way, where now psi is a vector collecting all these currents. And so, just a sec, so we get to a closed dynamical description in terms of the terminal species only. Yes.
The quasi-steady state assumption that you make, I can imagine that to be valid for, say, the external species that have a chemostat. But if there are some species that are getting shared by some other module, do you think the quasi-steady state assumption of them being large be valid there?
Well, it depends on the abundances, but like if you think that the species inside are enzymes, I mean, it's not unusual that the enzymes, the concentration of the enzymes is lower than the concentration of the substrates. And if you have this difference in abundances, then you get a timescale separation, even if the terminal species change in time. And I mean, with simulation, you get a wonderful timescale separation just, I mean, with 1 order of magnitude in difference in the abundances. With just one order of magnitude in difference, I mean, you don't distinguish the two of them, basically, in concentrations. Okay, so just like for the module here, okay, this is the stoichiometric matrix, exactly the same as Massimiliano used during the previous classes. If you want, these are the two cycles, right? And if you multiply each cycle for the sub-stoichiometric matrix of the terminal species, like yesterday Massimiliano was multiplying it by the sub-stoichiometric matrix of the exchange species, you get the stoichiometry of these two reactions. Let me show you the cycles again. So this is the first cycle. Okay, this sequence of reaction, you start from here, you go here, and you go back. So the abundance of the internal species doesn't change. But the net effect is that we take 1F, we follow the cycle, and we get out 1W. So the net effect is a reaction interconverting 1F into 1W. The other cycle is this one. You start from here, you go here, here, and here. So the abundance of the internal species doesn't change, but we take in 1 Na and we get out 1 Nb. So -1 for Na, +1 for Nb. And the point is that we now have a full description in terms of the terminal species only. And for the module here, if you want, there are two reactions with two currents, which are not actually independent, meaning that each current depends on the abundances of all terminal species. And that's the reason why we drew like this, like black line, coupling the two together. Because the internal structure shares enzymes, and so the two currents of two reactions that look independent are actually dependent, okay? And these are, if you want, the two, like, the four rate equations for the four species. And in this case, INA and INB, they represent the effects of the rest of the network that we have hidden away, while for F and W, IF and IW describe the exchanges with the environment, whatever that is. Now the question, yes?
Francesco, could you quickly explain like where the CMs will come from and then how will they feed in the last
CM?
Yeah, the C gamma M's. That will depend on, that will tell you what's happening, how your internal cycles are being distributed, how your internal fluxes are being distributed between different emergent cycles.
Yes.
How will they go into the final equation? Or
These?
Yeah.
You plug it in. You assume that the internal species have relaxed to a steady state. So this is the expression of the current must be evaluated for the concentration of the terminal species and the steady state concentration of the internal ones. If you, this current, since this is a steady state of this reaction, can be written in terms of cycle.
Yes.
And then just plugging the expression of that current in the rate equation for the terminal species.
So the CMs will, the C's will also depend on the external fluxes, right?
The psi. So the psi, so C, they are just vectors, you know? The right null vector of the stoichiometric matrix.
Yes,
so they are independent. Psi, they depend on the concentration of the terminal species. They are the currents.
Mm-hmm.
And now I'll show you how to compute them. Okay. Okay, so the question is, okay, we started, if you want, with the elementary structure of the module. We found this way to simplify it to effective reactions between the terminal species, and we ended up having these currents that depend on the concentration of the terminal species. But how do they depend on the concentration of the terminal species? Okay. Well, there are different ways to compute them, to compute how these terminal species depend, these currents depend on the concentration of the terminal species. So there are two ways to compute them. I don't want to enter into the details of the two equations. It's not very interesting, but I'm trying to give you like the understanding of how the two equations can be used to derive the expression of these effective currents. If you are interested, we can discuss about how these equations are derived, but it's just math, okay? So the first equation, the assumption is that we are actually able to compute the internal steady state of our module, okay? So either we are able to compute the steady state of the module analytically or computationally. The idea is that we compute it for different values of the terminal species, and there's a transformation with some matrices, okay, which allows us to relate the steady state currents to the currents of the effective reactions. But as you can see, it requires us to know the internal structure of the module. So it's a little bit inconvenient if we don't know it. The other way is by measuring. So the idea is that you have your module. In some way, you are able to extract it from the rest of the metabolic network. You put it in a reactor, and you measure the currents, keeping the concentration of the terminal species constant. And you do this for different concentration of the terminal species. And then there's a transformation from the exchange currents that keep the concentration constant to the currents along these effective reactions. So it's really like I take the module, I put it somewhere, I couple it with now only things that I control. I fix the concentration of the terminal species. And if we are out of steady state, the module is interconverting the terminal species. But the external processes are those keeping the concentration of the terminal species constant. If I measure those currents, I can use a transformation to get back to the currents of the effective reactions. So I have to measure this for different concentrations of the terminal species to reconstruct how these currents of the effective reactions change with the concentration of the terminal species. This is just the equation that relates the two. Yeah, this is a, see,
all the cycles?
Yes.
Okay.
C is a matrix of all cycles. If like, okay, for the physics part of the audience, if you want to see, is the matrix with cycles. And this is just a projection of the all currents on the space of cycles.
Yeah, exactly. I was going to say it looks like a projector.
Yes. No, sorry, it is the matrix encoding the stoichiometry of the effective reactions. What? Yeah, which is not linear in P. Yes, because this is the rate equation. So, sorry, I can stay here. This is the rate equation, right? You have a stoichiometry times a current. The current is a highly nonlinear function of the concentrations, psi, and I is a high nonlinear function of the concentration. But this is just because like the current is accounting for how many times a reaction occurs, and the stoichiometry is giving you the variation of the molecules along that current, along that reaction. And that's the point. The idea is to measure them. So you put them in a reactor, you control the concentrations, and if you are lucky enough, you can measure the currents. And if you want, this is the equivalent of what you do with electronic circuits, right? You take a device, you apply voltages, and you measure the currents. And of course, if you cannot do that, it's a mess. And if you want, for the specific network we have here, this is the specific relation between the reaction currents and the exchange currents. Okay, so every time Na is consumed by the reaction producing Nb, the current INA must replace the molecule that I just consumed, and the current INB must take out the species that I just produced, and the same for F and W. Okay, now imagine we do that for each module here, okay? So we have equations like this for each module, and then we couple them together in a single rate equation for all terminal species that we had in the original network, but now the dynamics is described only in terms of these effective reactions. Terminal species only. So, sorry, I wasn't clear. So the point is that we assume that this the internal species reach a steady state. So the concentration of the terminal species is determined by the concentration of the of the internal species is determined by the concentration of the terminal species. Now, if I evaluate the current, it depends on the terminal species, on the steady state of the internal species, which is a function of the terminal species only. I express this current in terms of vectors, and the coefficients must be a function of the terminal species, because the function, the current of the elementary reaction is a function of the terminal species only. Now I understand. Here, why this is only a function of the terminal species. Okay. Yes. Yeah, it's not written there, but yeah. Because like the idea is that I measure these currents for a specific abundance of the terminal species. So I have the value of the currents of the effective reaction via these equations for that value of the terminal species. Then I need to change the values of the terminal species. So I measure the exchange currents for a different value of the terminal species. And via these equations, I find the value of the currents of the effective reactions for the new value of the concentration of the terminal species. Then I have to redo that again and again and again. Like with an electronic device, I measure the current for a specific voltage, I change the voltage, new current, I change the voltage, new current, etc., etc., etc. And I reconstruct how the current changes with the voltage. In the regime you are interested to, of course, yes. Like hopefully, yes. Yes. That's the hope. Yes. That's the idea. And if you want, yeah, I mentioned this here, but I didn't comment on it. Like now there are these synthetic chemists who develop these membrane reactors where you can put your system into like a sort of box. You have a semi-permeable membrane and you let flowing, like if you want for that example, a pristine solution of fuel at a given concentration. And this equilibrates very quickly with internal, with your system. And if you were able to measure this current, you could do exactly what I was trying to explain. Because you here, like you have like a pipe, let's say, you pump in fuel at a given concentration, you have a semi-permeable membrane, so you have an exchange and they equilibrate. The concentration here out, if you want, is equal to the concentration in. And so this is a way you control the concentration of the fuel inside your membrane reactor. And then you change the concentration out, it equilibrates, and you repeat again and again and again. And the point is that, yes, you need to be able to measure that current. That's the challenge.
I have a question. So the experimental strategy, the equation that you have written, so what does it mean exactly? Like I can visualize like experimental data, yes, and then the data is written in the form of an equation, or the equation basically captures the dynamics of the data, and that you're trying to fit to an analytical or computation,
no,
or what
you don't have to fit. So the data gives you the vector of all the exchange currents for each possible concentration. And this, if you want, again, it's just a projection from the space of exchange currents to the space of currents of the effective reactions. So this is given by the stoichiometry of these effective reactions, which for now, I'm assuming is given. The experimental data must collect the exchange currents. I will comment at the end how to obtain the stoichiometry of the effective reactions if you don't have any clue about the internal structure of your module.
So this prescription doesn't necessarily distinguish between the exchanges that are truly external to the cell versus the exchanges that are somewhat internal to the cell. Right?
I mean, like, here I'm imagining that I'm taking my module out of the cell.
Right.
I put it in a reactor, And so also the exchanges that were internal to the cell now become something that I'm in control of. So if you want, like before, these were exchanges with the rest of the network. Now I'm doing this thought experiment, if you want. I'm taking the module out. I'm taking my glycolysis out of the rest of my E. coli. I'm putting everything in a reactor and I'm measuring it.
Okay.
I mean, I understand that it could sound like, you know, science fiction, but
no, that's fine. I guess I'm just trying to anticipate a little bit how to sort of think about this. So is it that, so the stuff that's F and W, that's going to stay external once you take this module back and put it into the cell. But NA and
then
NB
they
are
will
going to
become coupled to the rest of the
correct.
network.
So are we going to, I mean, is the idea that we should have in our mind is that eventually we're going to have to find a way to self-consistently match all of those things among all of the modules?
I mean, technically you don't need that.
Okay.
Because here, I mean, because of this time scale separation, whatever is changing NA, it's kind of slower compared to what is changing the internal structure. So here it's me playing around with exchanges in this science fiction.
Just to figure out the characteristics. I see.
Yes.
Got it. Got it.
No. It's everything but mass action. Yeah, highly nonlinear.
In this case, in this specific example that you showed,
so I can compute them. I can compute them for this module, for all modules, but this one. So, and okay, for example, like, I mean, I don't remember them by heart, but like for like the, let's say for this module, okay? Let me write it over there. So the reaction, see, B, it's something like, I'm sorry, I don't remember them by heart. I should derive them, but it's something like F minus W. Here there are like products of the kinetic constants of the elementary network. And then also in the denominator, yeah, you have something like F plus W plus NA plus NB. I mean, they are all multiplied by some kinetic constants. If you want, the expression is not so different from a Michaelis-Menten one, just more complicated because you have more species. But the idea is that. Just I mean, I don't. For all modules, but this. Here, for example, we computed it numerically under the assumption that we know the internal structure.
Yeah. Maybe explain why, but that one.
Picky. So when you fix the concentration of the terminal species, all modules, but that one are linear. The dynamics of the internal species is linear. So we can solve it analytically. For this module, the internal dynamics has biomolecular reactions between M and M star. So in that case, there's no general analytical solution.
So just to understand, these folks are the fact that you have 2 M star going to A2 star. Is that how
yes, yes. So even with mass action kinetics, it's like.
Okay, so then you get a quadratic.
Yes.
I see.
Okay. So we have the full dynamics, right? If you want, for this network, this is the full stoichiometric matrix, which looks huge, but it's way smaller than the original one, right? We have way less reactions, way less species than before. And these are the only species that we are now tracking down. All the internal species have disappeared from our mind and our heart. Now the question is, okay, we did all of this, but can we still compute the dissipation, right? Because we are hiding away reactions that are elementary, reactions that dissipate. So according from what Massimiliano taught you yesterday, the answer to this question should be no, we cannot do this. The answer is actually yes, because otherwise I wouldn't be here talking. We can actually prove that the dissipation evaluated by accounting the contributions of all the elementary reactions is exactly the same as the dissipation that we would write by taking the delta G of the elementary of the effective reactions times the currents of these effective reactions. And the reason for that is this quasi-steady state assumption, because of what Massimiliano was showing yesterday, right? At steady state, the only things we care about to compute the dissipations are the cycles. And thanks to this quasi-steady state assumption, it's like our modules are interconverting the terminal species only through the cycles. And so a little bit of math doesn't matter. We ended up with accounting for the full dissipation, but just tracking down a very smaller subset of reactions. And the point is that if you want to, it's not like we are taking some reactions, we are putting them in a closet, and that's it. We are changing the representation of the modules from the elementary reactions to the effective reactions. And this transformation, this is not just hiding away reactions, allows us to track down the full dissipation, because these emergent cycles are the pathway dissipating at steady state. Right? This is just the same kind of math Massimiliano was using for the steady state. This is just that here he holds also in this quasi-steady state assumption, because in a way, the internal structure of each module is at steady state, relaxed at steady state almost instantaneously, while the terminal species are changing in time kind of slowly. Okay, and let me stress again, from all reactions that we needed to compute the dissipation using, the standard formulation, we ended up with an expression for the entry reduction which accounts only for these effective reactions. Okay, that's the remarkable simplification of the problem, assuming that, of course, you can simplify the modules.
Just to clarify, if I understand correctly, so if quasi-steady state, if I understand in the most simplest term, does it mean that the internal species attain steady state very quickly, but the
internal to the modules, NA, ND, NB, NX, blah, 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 all the others, they change in time.
They change. So that is the intuitive meaning?
Yes.
Of, okay.
It's exactly that.
Okay, so if I imagine this whole black box to be a cell and so many modules running inside a cell, so there are individual modules which internally attain steady state, but species which can interact between modules have not attained steady state because they are under constant, source. They get source from outside, and they like the fuel comes from outside. The waste is again put out of the cell.
And also, like if you think of S as glucose, of the blue module as glycolysis, you get out the pyruvate.
Okay.
And the pyruvate can change in time. And then the green box is like the TCA cycle and so forth.
Right.
And I'll try to show that.
Okay, got it. Thank you.
The time we have left. Yes. Well, the species that are exchanged, then they are buffered. But the internal species, I mean, you can have an initial condition, which is not a steady state. And so the internal species, the internal terminal species, like here, NA, ND, NE, NB, NX, they change in time until they reach steady state. If the external, yes.
Yeah, Francesco, if I can just emphasize that, of course, the full system can also be at steady state. And in that case, everything is exact. So the time scale separation is only needed if we care about the dynamics of these terminal species. So the theory is valid also in that case with that time scale separation assumption. But if you anyway care about the global steady state, the theory is exact.
Okay. And if you think about you can think about like the balance equation also in terms of a Kirchhoff current and voltage law, meaning that at steady state, you can rewrite the rate equation in a way that basically is telling you that the sum of all processes, whether they are exchange currents or effective reactions, changing the terminal species, well, they sum up to 0 at steady state. If you want, this is kind of trivial. The other result is that the delta G of the reaction, if you find an internal cycle of these effective reactions, the delta G along the internal cycle, if you sum up all the delta G of all reactions along the internal cycle, must sum up to 0. Okay. This is true also for internal cycles in general. It's just that you're not changing anything. So if you start from something and you end up to the same thing, you don't change the energy of your system. And here you have, this was designed to have an internal cycle, and along the internal cycle, the delta G of all these effective reactions sum up to 0. Okay.
Just to confirm, this voltage law is almost by construction, right?
Yeah, because you start from something that must satisfy both of them if you want. They are emerging like properties, but they are from construction.
Yeah, sure. The first one is just steady state. The second one is, well, C's are the right null vectors.
Yes.
Yes. Okay.
Of the whole matrix.
That's right.
Okay. Now, if you want, we simplified our initial problem to something like this. Okay. These are the same reactions as before. I just didn't draw the boxes, the modules. Okay. The thing is now, because of this kind of coarse graining, these reactions are not elementary. They are effective reactions which don't follow mass action kinetics. Now you can basically repeat the same thing as before. If there's a time scale separation between the species that are exchanged and the internal ones, you can go from this description to a modular description of these effective reactions. Again, if you have this time scale separation, or as steady state, this is true always. Okay, so it's a way to say, okay, I have a module, sorry, I have a network. I construct modules, I simplify this. Now, if I have different networks that I simplified in the same way, I can further coarse grain my system and create, if you want, a second order module. And to answer the question that was asked to me at the beginning, if you think about this module, you can coarse grain it, or you can split it into something like this. So a module interconverting only Ne into Pe, because the enzyme here, let's think of it as an enzyme doing that, is unique for this reaction. While the enzyme, if you want, interconverting NX into PX and NB into PB is the same in both cases. So you cannot split this module into two submodules. You cannot do that because the currents of these two reactions are coupled together. If you want, like here, where the current of the reaction F into W depends also on the concentration of Na and Nb. The reason for that is that the enzyme doing that, the internal species that promote that reaction is the same for both. Okay, one last point, then I go to the application, I swear. Now, I did everything assuming that I knew the internal stoichiometry of the reactions, right? I constructed the effective reactions using the cycles of the internal reactions. But what if we don't know the stoichiometry of the internal reactions, right? Which is, I mean, a reasonable problem. I'll show you how to do that for just the example. I won't give a general answer to that. So the idea is the following. Again, you need to do something maybe, I don't know, absurd. You need to take your module out of your system. You put it again inside the reactor. And now the thing is that instead of starting exchanging all species together, you start exchanging all possible pairs or combination of species, and you measure the currents. Now imagine that you start exchanging only F and W, F and Na. Okay, so you are in this reactor and you couple it to something that exchanges only F and Na. Okay, and you wait for the system to reach a steady state. Now, from what I told you before, there are no effective reactions interconverting F into Na. So sooner or later, since these two species are not coupled, your system will reach a steady state where the exchange currents are zero. If we want to use the language Massimiliano used until yesterday, F and Na, they break independent conservation laws. Each one of them, once exchanged, break a different conservation law. So the system doesn't have a cycle here. The system will reach an equilibrium, which means that there are no effective currents interconverting one into the other. Now I take another possible pair of species, let's say F and then B. I wait for the relaxation to equilibrium, to steady state. I need to wait because now I have also the terminal species inside my system that need to relax. And also here, if you want, they are not coupled via an emergent cycle. Both species break two independent conservation laws. So sooner or later, I will end up with a steady state where the exchange currents are zero. Now, if I finally get the right pair of species, like F and W, and I wait for a steady state, I will end up with something like this. So I will see that for every possible concentration of F and W at steady state, the current injecting F is exactly minus the current extracting W. Or if I'm injecting W, I will extract F, which means that whatever is going on inside my system is interconverting one F into one W. And the stoichiometry is 1-to-1. This is the reaction. If I now take the other two species, again, sooner or later I will reach a steady state, and I will see that at steady state, the current of, let's assume, injection of Na is exactly equal to the current extracting Nb, because our effective reaction is something that takes one Na and gives out one Nb, 1-to-1. So the currents are equal except for the minus sign, and the stoichiometry is this one. And now we completed the set of species that we can exchange. So we have the full set of reactions. If we didn't, we should go on with further combination of species until we complete the set of reactions. Okay. Yes, question. Yeah, we are at the deterministic level.
Principle, if you did have, there is no guarantee that if you measure the current of one and the current of the other, you'll always get it to be zero. The stochasticity can induce non-trivial currents.
I mean, the stochastic version of this is a work in progress.
Sure, I accept that. Another one was 2 slides before you said you could do the repetition at higher level. I mean, you could do modules upon modules. I mean, it's basically building like a renormalization group.
Again, sorry?
Like a renormalization group transformation. If you go higher and higher. So I wonder if there are notions of fixed points and you can classify things into irreducible reactions.
I don't know. I don't think so, but I should think about that.
No, because this is basically a set of dynamical equations, dynamical systems, and you're co-screening over slow variables and looking at effective equations for the fast variables.
But the effective equations are highly nonlinear.
Sure. But I'm assuming that you can do that maybe computationally, or numerically, or in some perturbation calculation. Can you classify them as effective? Like you said, you can't break up that into 2 parts. I mean, there's a current coupling. Yes. Exactly, right? That's an irreducible part.
Yes.
So I wonder if this connects to things like motifs that people know in biochemical networks.
I honestly don't know. I don't know if you know. Key. So I hope I gave you an idea of how we were trying to go from a description tracking down all elementary reactions to something that is more complicated and requires coarse-grained approaches that don't track down all these elementary reactions while still, in a way, recovering at a good level the information about the dissipation. Okay, now let's try to apply that to central metabolism of E. coli. Okay, so we assume that our system is composed of only glycolysis, TCA cycle, and electron transport chain. Okay, everything else is the environment. Okay, and the point is to apply the same kind of reasoning that I showed you until now to the central metabolism of E. coli. And spoiler alert, you will end up with just one effective reactions interconverting the species that enter this metabolism. But let's try to do that step by step. So again, we consider glycolysis, TCA cycle, and electron transport chain. And they are kind of our 3 modules, okay, with the same language as before. So first module, we need to characterize it, and assuming that the species that are exchanged with the environment are these: glucose, oxygen, ATP, water, CO2, H+, phosphate, ADP. Right? These are the species that are exchanged with the environment. Everything else is internal. And these are, if you want, the terminal species of the modules: glycolysis, TCA cycle, and electron transport chain. Pyruvate, NADH, NAD+, and I have no idea how to call them. Sorry for the biologist. Anyways, so first module, glycolysis. So we have some species that are those that exchange with the environment. And then we have the species that are the other terminal species that are internal to this system we call E. coli. Okay, so if we use this approach of determining the emergent cycles, I won't go into details because I think that maybe the result is more interesting than the details. But in the end, you will end up with just one and only one effective reaction between all these terminal species. Okay. However, we can split, for those of you who were here also for the tutorial yesterday, we saw that sometimes we can split an effective reaction in basically 2 processes, right? Because we can split this reaction into 2, if you want, effective processes which are mass balanced, meaning that all the atoms on the left are balanced by the atoms on the right. And so we can read this effective reaction resulting from the emergent cycle as the sum of 2 processes. One is basically the decomposition of glucose into pyruvate, and the other is the interconversion of ADP into ATP. Now, if you think about writing the second law for just this module, like yesterday in the exercises, we can write the entropy production as the sum of 2 contributions which are coupled by the same current, because it's just one reaction which we are splitting into 2 processes. So if you want, the general expression, which is the delta G of glycolysis times the current of this emergent cycle, can be split into the sum of the delta G along the combustion, if you want, of glucose, consumption of glucose, times the current of the glycolysis, minus the delta G of the production of ATP times the current. And we can define an efficiency by comparing the ratio between these 2 work sources, assuming that one is positive and one is negative. And this doesn't depend on the current, because the current is the same. We have just one reaction, and we are splitting the reaction into 2 processes which occur with the same speed. So when we compare them, the ratio, if you want, here you would have the current and the current. But since the current is the same, they cancel out. And we ended up that we are able to compute the efficiency of glycolysis by only taking care of the physiological concentrations of the terminal species. So we don't need any information about the dynamics to look at the efficiency of glycolysis in E. coli. Second module, if you don't have any questions. Second module. TCA cycle, same logic. We have these terminal species, right? Some of them are internal to the system we call the central metabolism of E. coli. Some of them are exchanged with the environment. If we repeat the reasoning I was trying to explain, so we computed the emergent cycles and blah, 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 we end up with, again, one emergent cycle whose corresponding reactions in terms of the terminal species is this one. So you go from pyruvate to, how is this called? Coenzyme Q, H2. And DH and blah, 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 while interconverting ADP into ATP. Okay? Again, one reaction, but we can again split it into two processes, which are mass balanced. One, accounting for the interconversion of pyruvate in the, I already forgot the name, this one. And the other, which represents the interconversion of ADP into ATP. So we can split again the second law that, yeah.
This assumes steady state, right?
Well, I mean,
no. So like just naively thinking about it, now you have a mutation in one of your you split your effective reaction in two, right?
Yes.
And now you have a mutation in one of these enzymes, right?
Yeah, then no.
Then your current's going to change for one, and they're not the same, right? And then probably it's going to accumulate or something. So it's steady state.
No. So the internal species are fixed.
Yeah.
So the enzymes there in this analysis are there and they don't change. So if you have like a mutation or whatever, you break down everything. So and it's the reason why, if you want, we are still not able to like couple this with like regulation, genetic regulation, like when enzymes are produced, et cetera, et cetera. Here we are assuming that the enzymes are just internal to the modules and they don't change, of course. Yeah.
So also from the TC and the ETC, there's all these anabolic reactions that are happening, right? Does that not contribute to?
I will couple the gluconeogenesis to glycolysis after this.
Okay.
Yeah, for now, we just assume that our system is made of glycolysis, TCA cycle, and electron transport chain. Okay. So everything else is not there. Okay. Of course, this is a simplification, but like it's a way also to illustrate how this can be applied. Okay, if we repeat this, so we split one single emergent cycle, one single effective reaction into two processes, and we write down the second law in this fashion, we can compare an input and an output again. But since it's originally one single emergent cycle, one single effective reaction, the current is the same. And by taking the ratio between the two work, the input work and the output work accounting for the production of ATP, this doesn't depend on the concentration, on the currents, and we can evaluate it by just checking the physiological abundance of all these terminal species. So again, we don't care about the dynamics. Here we can evaluate the efficiency just looking at the concentrations at steady state. Final point, the electron transport chain, our last module. Well, in this case, if we consider these species as terminal species, we have two emergent cycles, two effective reactions represented by this reaction and this reaction. So the entropy reduction rate, the second law, is given, the dissipation is balanced by two contributions representing the two reactions, which are now running at different rates. So to evaluate the efficiency of the electron transport chain, we can actually we need to consider the dynamics. We cannot use only the physiological conditions. The reason for that, again, is because the two emergent cycles can occur at different rates. But if we put everything together, so we have one effective reaction for glycolysis, one effective reaction for the TCA cycle, two effective reactions for the electron transport chain. But if we now consider these effective reactions and we only want to look at the interconversion of the exchange species, we ended up with just one effective reaction, which is given in terms of, so the glycolysis occurs one time, the TCA cycle occurs two times, this emergent reaction of the electron transport chain must occur 10 times, and the other one 2 times. If you want, this is your final emergent cycle. And since, again, you have just one emergent cycle, okay, the second law can be written in this fashion as the time, so the energy reduction at dissipate is given by minus the delta G of this single effective reaction, times the corresponding current. But we can split it again in a reaction accounting only for the combustion, if you want, of glucose, and another accounting for the production, for the interconversion of 26 ADP and into 26 ATP. Let me stress this once again. We can do this splitting only because the two processes we get out of the splitting are mass balanced, meaning that all the atoms on the left are exactly equal to all the atoms on the right. If this wasn't true, we couldn't do that, okay? So we can split again this term in the second law as the sum of the combustion of glucose and the production of ADP. One in physiological condition is positive, representing the input power. The other is negative, representing your output. You compare the two, because they represent the efficiency, as also Massimiliano explained yesterday, and the result doesn't depend, again, on the current of this emergent cycle, because it's unique. So we can evaluate the efficiency of glycolysis, PCA cycle, electron transport chain, without caring for the dynamics. We just care about the abundances, right? Now, is that all? No. Because the point is that if our system is it includes gluconeogenesis, okay? Now the point is that if we have glycolysis and gluconeogenesis, what we get out of our analysis of that module is that we have actually 3 emergent cycles and 1 internal cycle. And so we should sum up the contribution to the dissipation due to the 3 emergent cycles, which again can occur at different rates. And if we again extract, these are the different rates of glycolysis, gluconeogenesis, and the futile cycle that we have here, which is not interconverting anything in the end. But so if we rewrite it again as a contribution and an output power accounting for the interconversion of ADP into ATP and an input power, we are left with something that depends on the currents of all reactions. So if we look at the efficiency is going to depend on the dynamics. So we cannot get rid of the dynamics in this case to evaluate the efficiency of this process. So we need to track down also how species change in time. If you want, the rate at which these effective reactions occur. And I mean, one option could be to use this idea of measuring the currents of these modules, hopefully experimentally, otherwise it's more complicated, of course. So in the end, okay, I was faster. So I hope I was able to explain which kind of approach we developed to simplify a complex chemical reaction networks into modules coupled together, and kind of show how this becomes very useful when dealing with metabolic networks. Now, for now, we applied it to central metabolism of E. coli. We also played around with the coupling between calcium dynamics in the cytosol and the efficiency of mitochondrial metabolism in eukaryotes, but this is basically it. And however, if you want this approach of creating modules and characterizing modules in terms of a sort of current concentration characteristics, there's no clear reason why this cannot scale up and be used to basically go from something like metabolism to studying the energetics of full organism to maybe something that is like a food web, where you have trees harvesting energy from the sun, being eaten by animals, eaten by other animals, eaten by bacteria when they die, which then becomes the sort of food also of plants, and also at the level of ecosystems, etc., etc., etc. Right, last slide. I mean, we presented a specific, we tried to give a complete and systematic description of how to construct a thermodynamic theory on top of a dynamical system, right, on the blackboard. And then we try to show this application to a circuit theory, which is basically this paper which builds up on previous papers on the coarse graining of chemical reaction networks. And then we study, I try to show today the application to metabolism for E. coli, which is this paper. And then if you are interested, there's this other archive right now on this coupling between calcium waves and TCA cycle. But the group of Massimiliano in Luxembourg really works a lot in the last more or less 10 years about these thermodynamics of chemical reaction networks. So if you want, the theory extended to beyond the mass action kinetics is in this paper. And this is for homogeneous systems like the one we discussed during the course, but we can deal with reaction diffusion. We can deal also with the problem of like how to characterize information exchanges between subnetworks, photochemistry, and then molecular motors. And that's basically it. Thank you.
I had a question. So when you said effective stoichiometry, so I'm just trying to understand that term. Now, if it's metabolic reactions, it's properly balanced, like glucose has 6 carbons, so the output will also be balanced with 6 carbons and so on. But the one you mentioned about the calcium signaling that you have modeled, so I can sort of think, correct me if I'm wrong, I can sort of think of that signaling as a, calcium being a secondary messenger in the cell. So it can probably activate a couple of proteins, and then that can influence some activities inside the cell, okay, like cell proliferation or any other activities. So how do we think about stoichiometry in that case? Even if you want to write the equation for that, like it won't be like, 10 calcium plus something. So how do we go? Is that what is meant by effective stoichiometry when you?
No.
Okay.
So effective stoichiometry is like the example I. So the notion of effective stoichiometry, if you want, is this one. So I look for these cycles, right? And along these cycles, I can determine the ratio between how many fuel molecule I'm interconverting and how many into how many W waste molecules I'm interconverting. So it's really counting all the atoms, okay? So it's really, I follow the reactions, I take something in and I put something out, but they are mass balanced. So all the atoms in F are all the atoms in W. About the question on calcium, here. So what we did was like to consider the mitochondrion as our system, okay, and to study the efficiency of this object. And we had the rest of the model accounting for the dynamics of calcium. But calcium plays, as you said, a regulatory role. So it doesn't enter the stoichiometry of the effective reactions in the TCA cycle. It enters just the currents of these two effective reactions. So the calcium for us in this model and the way we treat it here is just, a species which is externally controlled and tempers with the currents of this effective reaction, but it is not involved in the reactions. So it doesn't appear in the stoichiometry of those reactions. But we needed another part of the model, which we didn't care about the energetics, but it was like the part of the model accounting for the dynamics of calcium.
I see. So now if you think of calcium activating a particular protein, so what would you think of what would be the assumptions or the, equations that will capture the activation of a secondary messenger, activation of a protein by a secondary messenger? Has it got to do something with Michaelis-Menten or?
I don't know how to give a general answer to that. In this case, so we had some collaborators. We were able to collect what was known about the modeling of mitochondria, and basically we had already we took from the literature models of the currents of these effective reactions, And already in that modeling, there was like an expression of how calcium changes the rate of the reaction. But beyond that, I mean, I wouldn't know how to answer in general. I'm sorry for that.
I have a question regarding what you might call the sensitivity of the system.
Yes.
So your output measure of how good the system is efficiency, which you calculated at the end. Now, how does this efficiency change generally when you tinker with one of the internal metabolites? So for example, someone asked about a mutation earlier. If there's a mutation which reduces the activity of a particular enzyme by half, how sensitive is the system in terms of the efficiency?
It's an open problem. It could change everything, I don't know.
Yeah, the reason I ask this is the not universal, but fairly general observation is that efficiencies of metabolic systems are not affected by enzyme changes up to 50% or even more. And this is the famous Mendelian so-called law of dominance. Can that be recapitulated here?
I guess, I mean, we should try. I mean, we didn't try.
That would
okay, let me try to give you like a complete answer. So in a way, if you want, we assume that we studied the efficiency of these processes on a time scale. The internal species don't change. Okay, this is an implicit assumption of what I presented. Of course, if they start changing, we should account for that. But at the moment, I don't have any like, you know, hardcore proof or like a guideline to give a precise answer to your question.
Thinking of answer where you would change the internal species and follow the change in efficiency. I'm looking for a before and after kind of situation. So you had one steady state at the beginning and you settled to a second steady state at the end.
Okay, so let's put it like this. For okay, let's say glycolysis. Right? Imagine that we change the abundance of some internal enzyme. Okay, then that wouldn't change a thing, because in the end, we have just one emergent cycle, we have just one effective reaction. And since the efficiency doesn't depend on the rate of that reaction, which is the thing that depends on the abundances of the internal enzyme, that efficiency won't change. The thing is, if we change the stoichiometry of the internal processes, everything changes. But if we just change the abundance of the enzyme, this is like the same.
And that's because there's only one emergent cycle.
Because there's one emergent cycle. For like the electron transport chain, you change the abundance of some enzymes, since the efficiency depends on the rate of the reactions, then you need to account for that. And I don't have a precise answer.
Only if
play a role. Yeah.
Otherwise, it won't.
Are coupled to what? Yeah. Are coupled to the changes, exactly. Yeah. I mean, but it's always a problem of what you call a system or not. And I mean, it depends on what you care about in a way.
But if I can make a comment about the gluconeogenesis example where you have 3 emergent cycles, it is known in biochemistry that these two things typically do not happen together. So they are regulatory mechanism such that for certain concentration of glucose, only glycolysis is activated and gluconeogenesis is shut off. And when glucose goes down, the opposite happens. And I think this is something we would like to understand better, but we are not, this is future work, understanding how this regulation can transfer flux from one to another emergent cycle, because the kind of naive assumption is that it's kind of useless for the cell to have multiple processes going in parallel. You typically want to have one in one condition, or at least one predominant one under a certain condition. And then if the condition change, you can have metabolic switches. So all this is still open and should be explored.
So I was thinking of the scaling up to sort of bigger systems argument that you were bringing up towards the end. And it seems like in this sort of prescription that you were showing, whether the modules, you have these modules that you draw, and then you have this bigger boundary of the cell that you draw. And then some of the exchanges between modules are internal, and then some of them are external. Right? But it seems like in principle, I can already think about two species by just drawing this big box around, and just breaking up these modules into two different species. It seems like without, yeah.
May you repeat this last part?
Yeah, so if you go to any of these sort of module diagrams that you're drawing, like where you show multiple modules. Yeah, so so you're also drawing this big black border around all of the modules, right? Assuming that some things are outside the cell. But I could just also imagine that there's actually two cells. One cell which has the two modules at the bottom, one cell that has the two modules on top. And then the cells are exchanging these NB, NE, and so on and so forth. And it seems like the theory just goes without any problem.
Yeah, it's the thing I was trying to explain, saying that you can go one order further. Like you can take whatever you call the system now, and now you can call it a module. So if this was like the entire organism, so the different steps, each arrow here represents each like step of your metabolism, then you can like consider a module, which is the full organism. Then you can maybe couple other organisms. So what you're saying, I mean, in principle, it should work.
Yeah, and I guess what I was trying to say was that it seems easier to have your co-screening assumptions be satisfied there than inside the cell. The reason being that it's probably more reasonable to imagine that different species or different cells are exchanging fewer things externally than internally, whereas in your modules, that's kind of what you want, right? So
yes.
So I was trying to say that there's simplicity as you sort of scale up somewhat.
Someone here would agree with you. I mean, yes, makes sense.
Comment on, so when you were just talking about glycolysis etc, you had one emergent cycle, but then you added gluconeogenesis, you had 3 emergent cycles. Can you comment on
because like there are more reactions. I completely changed the stoichiometry. So this is the stoichiometry of only glycolysis. If I look at, I have these other steps, these, I think, wait, yeah. I have these other reactions that are here, here. You see, they were not there before. I assume they didn't exist.
Okay.
Now, when I consider that they actually exist, then there are multiple pathways.
Okay, and
because,
yeah.
I mean, yeah, it's like Massimiliano said, right? I mean, since in most it seems that like there are conditions such that only glycolysis occurs and conditions such that only gluconeogenesis occurs. In the first case, we assume that our system is glycolysis, TCA cycle, and electron transport chain. But if we look more carefully and we plug in also the reactions, basically implementing gluconeogenesis, then there are more reactions and there are more effective reactions too. That's the point. It's always like where I put the boundary of my system, right? But if you include the gluconeogenesis, then you cannot compute the efficiency because you have more emergent cycles.
More questions? I don't see any. Thank you very much, Francesco. Very nice talk. Thank you, Massimiliano, both of you, for this very nice course.